Yeah, I'm a group leader at the Earlham Institute. I look after a group known for sort of historical reasons as the Technology Algorithms Group. We're a group focused on developing new methods in metagenomics, and we're particularly interested in situ metagenomics, taking the lab out to the sample and real-time analysis. And both of these things are things that are only really enabled by nanopore technology, the only technology that you can do this kind of real-time progressive analysis while sequencing continues, and the kind of only platform really compact enough to take out of the lab. I'm from a computational background as well as really after the physics that Mark mentioned, but the group also has wet lab expertise in it as well, so we kind of cover both sides. What I wanted to talk about today was really kind of two bits of work that are in pathogen detection, and they're kind of linked up by software really. So the sort of underlying story over the next few minutes is the sort of development of a software package really with these kind of applications as a sort of way into that. So I'm going to talk about some clinical pathogen detection work that we've done in the past and are continuing to do now, and then how that led to the development of this tool called MARTI, and then airborne pathogen detection work that is also really being enabled by MARTI. So first of all, the clinical pathogen detection. So I want to go back to a project a few years ago that we carried out with Lindsay Hall's group at the Quadram Institute, where we were looking at pathogen and AMR analysis in preterm infants. So these preterm infants are very prone to infection. They have an immature immune system and underdeveloped gut physiology, given prophylactic antibiotics often. And one of the things they're at risk of is something called necrotizing enterocolitis. This is a really serious condition. It's associated with gut microbial dysbiosis, so this increase in pathogenic bacteria and a reduction in beneficial bacteria. Number of pathogens linked to it, but it's kind of characterized by quite rapid change, and existing diagnostic tests are quite slow. So it's really key to work out what the pathogen is and its resistance profile, and this involves typical approaches of plating out samples and waiting for them to grow, and it can take 2 or 3 days. So together with Lindsay and a trial she was running at the time, we wondered, could we use nanopore sequencing for rapid surveillance and diagnostics of these patients? So the paper we published a few years ago, it's got kind of comparisons of Illumina and nanopore data, and it's got kind of longitudinal studies. But the thing I wanted to highlight, for me, the most exciting thing was the rapid sort of diagnostics proof of concept that we attempted. So we wanted to see how quickly we could go from sample through to complete analysis. So we set up the pipeline you can see here, where we go from fecal sample through DNA extraction, nanopore sequencing, and analysis. And for analysis, we created a new software platform called NanookRT. And the reason for this is that whilst the nanopore platform enables real-time analysis, it was actually poorly served at the time for real-time analysis tools. And we couldn't do what we wanted to with Metricore, which was the Oxford nanopore solution that was available at the time. So we created NanookRT. It performs classification of the metagenomic sample, so it uses a BLAST and lowest common ancestor approach for those of you that know about that sort of thing. So it assigns reads to taxa, and then it also does AMR analysis via the CARD database, the Comprehensive Antibiotic Resistance Database. And so NanookRT would analyze the sample as sequencing was happening. You get these kind of readouts like you can see here with a kind of taxonomic view or a donut kind of summary. There'd be analysis of the top AMR genes. And then it also did something we called walking out, where it attempted to look either side of the AMR genes to identify the enclosing host bacteria, and then would kind of give you readouts on that. So how fast could we do this? Well, these first plots are really just showing the difference between the data as you see it at 1 hour and 6 hours of analysis, and you see very almost identical results in these kind of patients the pathogens tend to dominate what you see in the gut microbiome, and it's very obvious within a few minutes of sequencing, not even an hour, what the pathogen is. AMR data takes a bit longer, so this heat map is showing different classes of antimicrobial resistance gene in the y-axis versus time, the top of the x-axis, or number of reads analysed, the bottom of that axis. So you see the most common AMR genes, multi-drug efflux pumps. You see lots of those really quickly. It takes a bit longer to get to perhaps the more informative genes like these Klebsiella pneumonia specific SHV variants, but the first of which coming after about half an hour. So we put this all together. This was the pipeline you saw at the start, and we can now put some times in here. So we receive the fecal samples at hour 0. We're sequencing within about 3 hours, and then we sort of argue in the paper within about 4 hours there's potentially enough information to prescribe bespoke antibiotics rather than sort of broad spectrum ones. And that, as I say, compares to maybe 2 or 3 days for a conventional process. So the potential for these kind of nanopore approaches for rapid diagnostics and for real-time analysis, we think we've kind of shown. And we started to see that this tool that was developed for a very specific application had uses beyond that specific application. And one of the key things about it is that it didn't require any kind of internet access. So that the Oxford Nanopore solution at the time required reads to be uploaded to a cloud service. And the fact that we could run it locally, actually just on a laptop if necessary, meant that we started to see these other uses. And we started to take nano-CAR-T out to beyond the clinic, and a couple of examples are shown here. So on the left-hand side is a PhD student called Emma Langen, who's finished now, but she went off with a British Antarctic survey on the research vessel you can see there, and she was interested in phytoplankton in the Southern Ocean, and she used nano-CAR-T for analysis. So she'd have this 2-laptop setup you can kind of see in the bottom there, where she'd use one laptop for nanopore sequencing and then carrying out real-time analysis on the other one using nano-CAR-T. And on the right-hand side is another trip that I took to Iceland with Sarah Stewart-Johnson, who's a planetary biologist from Georgetown University. She was interested in these kind of extreme environments in Iceland as an analogue for Mars, and interested in what kind of in-field sequencing and analysis we could do of extreme microbiome communities there. And so again, we use this kind of 2-laptop approach with a sequencing laptop and nano-CAR-T for analysis. So nano-CAR-T was starting to be useful, but we knew it could be better. It lacked a bit of flexibility in the back end, and it had quite a clunky front end. And so over the last few years, we've developed MARTI, and MARTI stands for metagenomic analysis in real time. You can see in this video, you can see it running. So MARTI takes the back end of nano-CAR-T, it's heavily updated, and then it adds this really nice web-based front end that Ned Peel developed initially as a PhD student and then more recently as a postdoc. So MARTI supports this same BLAST, lowest common ancestor type approach to metagenomic classification, but also adds a number of other tools, Diamond, Kraken-2, and Centrifuge. So Kraken-2 and Centrifuge are particularly kind of have lower compute requirements and can kind of run more easily on a laptop. It contains the same AMR analysis, but slightly extended. So you just saw scrolling past was a window of AMR gene hits, and then what we're looking at now is the walkout analysis approach that I talked about before for placing AMR genes in context. It can run in a variety of settings. So you can run it in an HPC environment where you run the back end on an HPC, and then you use a web browser on a laptop or a tablet for interacting with the data. Or you can run everything on a laptop. And obviously that's going to be more challenging if you use BLAST and large databases, but it's perfectly possible for smaller databases and things like Kraken-2. And then you can compare samples. That's what we're seeing here, various views comparing samples. You can kind of drill down into the data, view it at different taxonomic levels, set different filters, export images as vector images for presentations and papers. Marty's become a really useful tool for us. We published it towards the end of last year. If there's people here that are interested in nanopore metagenomics, we'd really love you to try it. You can read the documentation, you can try out the software. We've got a web server where you can go and interact with some of our data sets and try it out and see if it's the kind of thing that would be good for your data. And then to install it, we've got a GitHub, and it's also available on Bioconda for probably the easiest way to install it. So Marty's become really key to all kind of metagenomics we do. It's our kind of go-to platform for nanopore metagenomics. And one case where that's particularly been true has been our airborne pathogen detection work, or AirSeq as we call it. So AirSeq's our method for detection by DNA sequencing of biological material in the air. It's a long-standing collaboration with Matt Clark, who was previously at the Earlham Institute, now is at the Natural History Museum in London. Our sort of original focus with AirSeq was crop pathogens and the desire to have sort of more targeted treatments of pathogens in agricultural environments, and to be able to alert to the presence of emergent pathogens. So our vision ultimately was for a box that could sit in a field monitoring the air and alerting farmers or agronomists as pathogens reach threshold levels or new pathogens emerge. And then ultimately, the dream would be to have a network of these sensors around the country. So for quite a long time, we've been developing AirSeq, and we've got a really mature pipeline now that includes sample capture, DNA extraction and purification, nanopore sequencing, and analysis using Marty. We don't have a fully automated box that can sit in the field yet, but we've kind of automated pretty much each of the steps along the way. To give you an idea of what AirSeq data can give you, this is a sort of relatively early proof of concept, but we only published in 2024. And so this was actually back in when we were still using Illumina sequencing. Initially, we used Illumina sequencing, and we're now totally nanopore sequencing for this. And we carried out this trial in the fields behind the Earlham Institute, which were going crop trials at the time, wheat, barley, and pea trials. You can see our sampler in the field on the picture on the right. We sampled 3 times a week in duplicate for 2 months. So what you can see here on the left of the slide is a tree map that's showing all the samples over that 2-month period, basically giving you an idea of kind of what you can see in these samples. So we see a green box up here is showing insects. So the air collector we were using at the time did have a tendency to suck in insects from time to time, so we would see that reflected in the analysis. The ones we use now, less likely to do that. You see plants here, so we're picking up DNA from the crops, but also other plants in the area. You've got fungi and you've got bacteria as well. So we kind of see the things we might hope to see, but we were particularly interested in pathogens. So what Michael did, who led the analysis on this, was create a database of pathogen reference sequences based on Phibase. So Phibase is the pathogen host interaction database developed by Rothamsted. It's got details of pathogens and the hosts associated with them. So what you can see in this plot here is wheat, barley, and pea, and the red bar is showing the number of pathogens in Phibase, and the blue bar is showing the number of those that we detected at some point over the 2 months through our AirSeq data. And the heat maps at the bottom are giving a sort of more detailed view of some of those pathogens. But on the next slide, there's 3 that I've picked out as examples. So what you'll see is 3 columns here. This top row is showing the AirSeq detected level of the pathogen, and then we've got temperature, rainfall, and humidity data. So the crops are all sprayed at the start of the trial, so in each of these cases you'll see very low amounts of the pathogen at the start of the trial. This first example is the pathogen responsible for yellow rust. So we see it low at the start and gradually increasing in abundance over the time we're monitoring. And again, we see on the crops damage increasing over that time as well. The next one is barley powdery mildew. The lines of barley being grown were highly susceptible for this, and we see this coming back repeatedly during the time we're monitoring. And then the final example is quite interesting because it's something we see at very low abundance most of the time. The weather was very dry during that time as well, and then we get this peak in rainfall towards the end of the time. The plants are at that later developmental stage as well, and then this pathogen really seems to take off. And what we see in that data, and also in other agricultural data sets, we've done quite a lot of sampling in greenhouses, for example, is we can see the levels of these pathogens high in the air quite a long time before we see the damage on the crops, a couple of weeks before we see damage on the crops. And we can relate that as well to the environmental conditions as well. So you need the presence of the pathogen in the air and the right environmental conditions, and then we can see the evidence of the damage. We've also been interested not just in agricultural pathogens though, but in human pathogens in urban environments, and we had some DARPA funding where we were looking at this. So one of the things we did as part of that is to look at the urban air microbiome in London to try and understand the differences in the air microbiome around London. So we sampled at 13 different sites, and we did it twice. So we did a summer and a spring sampling 18 months apart. And what we can see is some clear geographic differences in the air microbiome. So what you've got here are the two data sets. So the top row is showing August 2019 sampling, and the bottom row is showing March 2021 sampling. And then on the left-hand side, we're just seeing a phylum level composition of these samples. So we see plants, fungi, bacteria, very small amounts of DNA from animals as well, but very small compared to the plants and the fungi. But what's interesting, the plot on the right is showing how unique each site is. So essentially the red is showing species that were present across all sites. The green is showing species that were present across most sites. And then you've got this set of species in blue that are specific only to that site, and then the yellow ones are species specific to only a few sites. So there's a common core across all sites, but there's interesting differences between the sites as well. And then the other thing we did is at one site we monitored weekly for a whole year. So this was in the Natural History Museum Wildlife Garden outside the front of the Natural History Museum. And you can kind of see these clear seasonal differences. So for example, in spring, the makeup is, sorry, I should say each of these bars represents a week going from summer 2020 on the left to summer 2021 on the right. So in spring, we see what we detect dominated by plants with all the pollen in the air. And then at other times of the year, it's fungi, it's Ascomycota, in the summer moving to Basidiomycota in the autumn. I've kind of run out of time, but hopefully you've got an idea of the ways we're using nanopore sequencing in these pathogen detection applications. All of the things that I've talked about are kind of ongoing projects as well. So the clinical pathogen detection, we obviously published that work with the preterm infants, but we've got a couple of projects going now with mothers of preterms with John Larty, who's here from the NNN, and also with Alison Mather. And so we're looking at as I say, infection in mothers and how that can affect the outcome for preterms. We're also, with Lindsay, Lindsay led a project that we recently pre-printed using the kind of similar kinds of approaches in patients with chronic liver disease, gut-related complications, in patients with chronic liver disease. So that was recently pre-printed using these kind of same nanopore approaches and using MARTI. And the development of MARTI continues. As I say, we'd really love to hear from any users, potential users. Feedback's very welcome. And then finally, on the airborne pathogen detection, we're still doing stuff with the kind of agricultural applications, and we're also looking at, I guess, biodiversity monitoring applications. And you can kind of see a little video running there of a project we've got where we're going to different habitats around Norfolk and North Suffolk, looking at marshes and broads and agricultural areas and the city, and looking at differences in the air microbiome. Yeah, all that remains is for me to thank all the people involved in these projects, and to thank you for listening.